Right, so today's topic shouldn't take the full hour, which means hopefully we can get it finished slightly quickly, which means that you guys can get away and enjoy your weekend. But it shouldn't be too difficult. I think it's just more about kind of understanding the construct and understanding especially the processes of mass transport. That's very important to kind of get your head around. Okay, so this one we don't really have slides for, so I'm going to explain it through kind of whiteboards and explain it through other kind of means, but we should still be able to get it done. Sorry, give me a sec. Just get it ready. Just still letting people in. Just give me a second. So, right. So today we're talking about mass transport in plants. This is again designed generally for the AQA spec, but there will be aspects of it that can be carried forward to other specs, especially OCR. So we'll start talking about Xylem, cohesion tension theory, phloem, mass flow, investigating transport in plants, and then evidence for and against the mass flow hypothesis. So Xylem and phloem are found within the vascular bundles in the stem. So often what you'll see is if you look at a cross-section of a plant, you will see vascular bundles located on the outside of the stem in sort of a circle. These are split into three different aspects of it. So it's got a xylem, it's got a phloem, and then it's got something called the cambion strip that runs in between it. And this strip is designed, and it's got stem cells, which are then used to grow the stem further. So xylem, phloem, and the cambion strip. But yeah, as you can see, this is your vascular bundle. Your xylem is on the inside, it's pointing in. Your phloem is in the middle, and something called the sclerenchyma, which is again your stem cells, it's your cambion strip, that then works on the outside. So xylem transports water in the stems and leaves of plants. Water, the first thing to it is it first has to be absorbed by the plant root hair cells, and then it is transported into the xylem and then transported to the rest of the plant. So xylem is usually long, hollow tubes made up of dead cells. The cells that make up the vessels have no end walls. So this is a continuous tube that water can travel through. So you can see here that there is only one-way flow. There's no end walls, one way only, and the outer cells are not living. They have been lignified. Lignified is basically, there's a substance called lignin. It's a polysaccharide, and it's deposited into the walls, and that kills the cells of the walls. That means that it also creates a waterproofing barrier, which means that the cells are waterproof so that there is no transport through from within the system to outside the system. But also lignification means that the xylem is able to withstand high pressures, which are very important for it to then be able to allow for flow to pass through it. The cohesion tension theory explains how water moves up the xylem. Cohesion tension theory generally explains something called transpiration. So transpiration, what topics will this session be about? I will go back to explain that. All around. There we go. So xylem, cohesion tension, phloem, mass flow, and investigating transport in plants, and then evidence for and against the mass flow hypothesis. So we talked about the xylem already. We're going to talk about cohesion tension theory, and then phloem, mass flows, understanding data. And then we'll end up with apologies, for some reason I seem to have disconnected. I will start again. So transpiration is the end, it's the evaporation of water from the plant, from the surface of the plant. Usually this is from the leaves, and it's from the front of the leaves, but we need to create a flow of water through the plant. So water needs to be able to flow through this plant because it needs to get from the root to the leaf. How does it do this? Well, it needs to be able to, one of the crucial things it needs to be able to do is it needs to be able to travel against the force of gravity, because gravity is designed to bring water back down, but it needs to be able to travel up the xylem. There are no protein channels in the xylem that it can be passed through. So the xylem needs to create a system that can allow water to move up. So the first way is, so this is by cohesion tension. The other way it's done is, first of all, through a negative pressure gradient. So I'll explain that bit a little bit. You've got the leaf at the top, right? And you've got the root at the bottom. If the water is evaporating from the leaf, that then means that the water tension or the pressure is going to lower in the leaf because there's less leaf, there's less water left. In the root, water is being pumped into the root at very high pressure. So the water pressure into the root is very high. This difference in pressure creates a pressure gradient. And we remember from our basic biology, everything in biology likes going down its pressure gradient. Everything likes going down its pressure gradient, which means that the water needs to go up the pressure and it needs to get up the root and it goes down the pressure gradient. That's the overall process. But how does it get up this root? So it gets up the root through cohesion tension. So cohesion is water molecules sticking together. This means that water molecules can stick together and create almost like, if you imagine kind of like a group of like small kind of water molecules sort of pushing themselves and shimmying their way up this surface. The other way is through tension. So tension is the water molecules sticking to the walls of the xylem. So you need both. You need the cohesion of water molecules to one another and the tension, which is the water molecules sticking to the surface of to the walls of the xylem itself. And both of these combined create cohesion tension, which allows for water to move up the plant. And again, this is again down its pressure gradient, which is important to it. So you can see that summary of cohesion tension. So water evaporates initially, that lowers the pressure. It lowers the osmotic pressure, which increases the osmotic potential. So lowering of the osmotic pressure here, increasing osmotic pressure here. But it still needs to find a way to overcome gravity. And the best way you can do that is literally the water molecules will kind of stick to each other and be attracted to each other up there, up the actual kind of xylem itself. Okay, so that's xylem and water transport. Does anyone have any questions about water transport so far? Any questions about water transport? Feel free to put them in the chat. I will wait for a minute or two, and then I'll answer the questions. So I'll start again at 25 past with any questions that you guys might have, and then we'll talk about phloem. Right, so we've had one question in the chat. Are we going to talk about SymPass and ApoPass pathways today? Yeah, I can talk about them. So let's go back. So the SymPass and ApoPass pathways both indicate the movement of water and molecules through it's mainly through the root themselves. So the SymPass pathway is basically movement of water through the cytoplasm of adjacent plant cells. The problem is it can't do that forever. Eventually, it'll reach something called the cambium strip, which is basically a part of the root that basically blocks movement through the cytoplasm. So everything has to move into the ApoPass pathway eventually. ApoPass pathway is movement of water through the cell walls or through the intercellular junctions between the cell walls. That's kind of it. It's not very complicated. You don't have to know a lot about it. Symplast, cytoplasm, apoplast, cell walls, everything goes into the apoplast pathway eventually. It just starts with the symplast pathway. You don't really need to know that much more about it. Okay, so what is phloem? Phloem tissue transports organic substances, including sugars such as sucrose and some mineral ions. Phloem tissue consists of living sieve tube elements that each have a companion cell within them. The companion cell is crucial for the process of mass transport. Sieve tube elements do not have organelles because they form part of the tube that transports the organic substances. Instead, the companion cells can carry the organelles, and the companion cells move substances into the sieve tube, and then that moves through, that moves the water through, moves the ions through the substance itself. Okay, mass flow. This is really important. So translocation is the movement of organic substances through the plant. Translocation moves substances from source to sink. So sources are the site of production, and sink is where the substances are stored or used up. Usually for a plant, a source is going to be the leaves of the plant because they're actively photosynthesizing. And the sink is going to be the root cells, because that's where plants store their organic material. Translocation is an active process, so it requires energy. Energy is required in the form of ATP, and there are 3 stages to mass flow theory. So the first stage, I will draw this out as we're going through. So you've got your sieve tube, you've got your companion cell, and you've got sort of movement through this. So, give me a sec. Oh, not too sure what's going on. Okay. Sorry, I think my computer's just dying on me all of a sudden today. It's not been great. Okay. So, the first thing you've got to do is you've got protons moving out of the companion cell by active transport. So these are actively being pumped out of the companion cell and into the what is like the intracellular fluid. So they're like the ICF, which is basically the fluid that exists between cells. Now, ions are pumped out here. This creates a concentration gradient for the protons. So there's low H plus here, and there's high H plus here, which means that protons can now move back into the cell by facilitated diffusion. And what are they facilitating the movement of? Sugars, like sucrose. So what this pumping, this active transport of protons out of the companion cell is doing is it's creating a concentration gradient of the same H plus ions for H plus ions to move back in by facilitated diffusion along with the sucrose molecule. That means that inside the companion cell, there's now going to be high amounts of sucrose. There's low sucrose in the phloem because phloem is flowing. So there's always a maintained concentration gradient. Now, quite easily, the sucrose can just diffuse by simple diffusion through the plasmodesmata into the phloem itself. At the sink, now, quite simply, how does this flow actually happen? You've got to be wondering. So this is the first step. Intracellular fluid, or it's this fluid between cells, is the best way to describe it. So now, how this sucrose, if it enters into the phloem, there's lots of sucrose, which means that the water potential goes down, which means water enters into the phloem. So water moves in. That increases the water potential, which means that it creates a pressure gradient, so that now there's flow happening. So flow happens because down the water pressure gradient, as usual, we've got the flow down the water pressure gradient. And it now moves to the sink. At the sink, simply put, the exact opposite happens, but not with protons. Sucrose can diffuse out because, again, there's lots of sucrose here, less sucrose in the cell. Immediately as sucrose diffuses out, it becomes used up or it gets stored. So the sucrose concentration gradient is always maintained as being low because the sucrose is always being used up, or the sucrose is always being kind of stored. That increases the water potential here, which causes water to move back out of the sieve, out of the phloem itself. And now you've got this recurrent cycle of this happening. And I'll summarize that with a whiteboard slide, as we did before. So as we've talked about many times, whiteboard slides are the way to go. This is kind of your stages of mass flow. And I'll give you a couple of minutes to go through that. And we'll start again at 35 past. So that's the end of. Yeah, that's the end of mass flow. So if anyone has any questions about that at the same time, feel free to put them in the chat, and we'll get started with the last bit, which is evidence in 2-3 minutes. Right, that's been a couple of minutes. So we've got a couple of questions in here. How does sucrose get stored as amylose and amylopectin if it's a disaccharide of 2 glucose? Because amylose and amylopectin are polysaccharides of multiple glucose. Are guard cells the same as companion cells? Yeah. In this case, I've made a mistake when I was writing this when I did my levels, and I've equated guard cells to companion cells. We are talking about companion cells here. Does assimilate molecules collectively refer to sugars and mineral ions? Mainly sugars, because again, mass flow is mainly through sugar. Remember, the xylem transports a lot of mineral ions as well. How is the structure of the phloem adapted to mass flow? Well, if we think about it, it's got the companion cell. The companion cell does most of the ion transport. Plus, it's got the sieve tube elements, which help with it preventing too large molecules from getting through. And it also helps maintain pressure in individual areas. So it means that sucrose, once it gets to the sink, it doesn't just leave again. That's the idea. The sieve tube elements basically help with compartmentalization is the idea. Right. We'll go back to class. Tracers and ringing experiments. So how did we prove that this was the way that things were happening? Well, we used radioactive tracers. So they were basically helped to track the movement of organic molecules through plants. Carbon-14 is a radioactive isotope of carbon. So plants can be grown in an environment containing radioactively labeled CO2, which is then incorporated into the organic substances produced during photosynthesis, like the sugars. Once it's then packaged into the sugars, you can actually see and follow by imaging how those sugars pass through the plant and where they're stored. This is through a process called autoradiography, which is basically placing cross sections of your plant onto the X-ray film and then watching the film become blackened if it gets exposed to radiation. How do they radioactively label CO2? They just they produce the CO2 in an environment where all the carbon that they're using is carbon-14, which means that it's different to the normal carbon. So again, they don't label it. They just make sure that it's being made from carbon-14, from radioactive carbon-14, if that makes sense. And again, you can watch that. The other way of doing this is something called ringing experiments. So in bark, on wooded stem, there is inside that bark, there is a ring of phloem. And on the inside of that ring is xylem. If the ring of bark is removed and the phloem is removed, you see a bulge of fluid will form above the section that was removed. The fluid, you can then drain that fluid, and you can see that that fluid is going to have a lot of organic substances comparative to the fluid below, which then implied that the phloem was important in transporting those organic substances around the plant. That's the idea. Again, you leave the xylem still intact, and if you leave the xylem still intact, it means that xylem then cannot be responsible for transporting organic substances. And you can see, remove the phloem, and you've left the xylem. Okay, I can go back to the slide about ringing. Done. Thank you. Cool. Again, this is quite easy. So recognizing relationships. So mass flow, supporting evidence. You've got the radioactive tracers and the ringing experiments both prove that mass flow happens in the phloem. Again, the structure of companion cells. So companion cells contain many mitochondria, which is then used to produce ATP. ATP is the essential component of active transport, which indicates that companion cells are specially designed to perform active transport. When plant stems are broken or cut, sap is released, which show that there is hydrostatic pressure within the tube. And when you release that, when you open the area, then the fluid is released. Solutes don't, evidence to object to this hypothesis is that solutes don't all move through the plant at the same speed, which they would if they moved via mass flow. Mass flow also suggests that sucrose should be delivered fastest to sinks with lower sucrose concentration. But actually, because it travels to all sinks at roughly the same rate, that implies that it's not being selective by just the gradient. There seems to be something else that is ensuring that it gets to all sinks equally. And sieve plates, by their very design, are there to impede the flow of, impede mass flow by creating an obstruction. So you've got all of these different examples of how kind of mass flow is obstructed through the substance. Right, let's do a quick pop quiz to end. So can people put in the chat what their answers are? C, we've got one, C, C. Let's get some more answers in. Let's double check, see if anyone else is saying anything else. D, we've got a D. Anyone else got any other answers? D, 2 Ds. Okay, so it's getting a bit tight here. 3 getting a D. Yeah, well done. People spotted that this is incorrect. Which of the following statements is incorrect? Not correct. So, yeah, the answer that's incorrect is D. Well done. Okay, next one. Which of the following statements is incorrect? Again, be careful about that. A, C, between C and A for some people. C, couple of C's. Let's find out. Yeah, it's C. Remember, xylem vessels to have no end walls, which allows for continuous flow of movement through the xylem, which ensures that the xylem stays, kind of the flow stays. Okay, last question, I think. Which of the following statements is incorrect? Let's give you guys a go. Again, put it in the chat. Yeah. Again, we're saying that translocation is the evaporation of water from a plant. No, transpiration is the evaporation of water from a plant. Translocation is the movement of solutes and organic substances from one part of a plant to another part of a plant. We'll skip these questions. But yeah, your crucial take-home points are xylem tissue transports water in the stem and leaves of the plant. Cohesion tension theory explains how water moves up the xylem. Phloem tissue or transports organic substances in plants, including sucrose, through the process of translocation. The mass flow theory can be split into 3 main stages and explains the mechanism of translocation in plants. Tracers and ringing experiments can be used to investigate transport in plants. Another crucial thing is tracers and ringing experiments aren't in every specification. So if you don't feel like you've covered those, you might not have. So don't worry if you don't feel like that's something that you've done. They're not in every spec, so don't worry about it. Okay. Does anyone have any kind of questions? I will be on. Otherwise, feel free to kind of wrap up. I know this is a quick one. Today is not too difficult. Tracers and ringing experiments are basically used to prove that the mass flow happens within the xylem. So tracers and ringing experiments show that not only does substances move through the plant from source to sink, but also it shows that that movement happens through the xylem itself. Any other questions? Otherwise, I'll wrap up for the session today. I hope to see you all on Sunday for chemistry, Sunday 6 to 7 p.m. as well. Chemistry session. Thank you, guys.